Right, so last week we talked about sort of basics of sensation and perception. We talked about some basic concepts, principles, foundations. We talked about a few sort of interesting methodological approaches. We didn't hit a lot of stuff in detail. It was really an orientation sort of process, but there was a ton of important information there for you. This week we're going to jump in and we are going to discuss a couple different sensory systems. First, we're going to talk about the chemical systems. This actually includes two sensory systems, right? Olfaction and gustation, both are chemical senses. Then we're going to jump into the body senses, talk about that for a little bit, and then we'll move into the lab activity. So both of these chapters are sort of short, only about 20 slides for each chapter, and then we'll get into that lab activity. Here are learning objectives. You guys can read those. Hey, who loves smell? Let's talk about that first, right? Smells great. It's actually, for humans, we actually consider it sort of a minor sense, right? People aren't using olfaction to navigate their environments. I mean, you could in some places, but I don't see people walking down 3rd Avenue going, wait a minute, yep, that smells like Harris. I'm going to turn in there. I suppose you could do that, right? Everything sort of has its own smell. A lot of things we don't even pay attention to. We have taste adaptation, smell adaptation. We'll talk about that. But there are some cool things that you can do with smell, and we'll talk about those. There's some evidence that indicates you can determine the sex of another person based on the smell of their breath or their hands. I don't know, that's kind of an interesting one, right? Right? I mean, you just smell people's hands, figure things out. Smell can also be used to diagnose illness, right? If you smell somebody and they smell like vomit, they probably got a stomach issue, right, Charlotte? I mean, that makes a lot of sense. It's a great diagnostic tool right there. I don't know, I've seen these studies where they train dogs to like smell like cancer cells. Have you ever seen those? Like, I don't know, I accidentally watched Dateline or something once and it was on there. There are also some interesting things with syncing up menstrual cycles, right, based on olfactory stimuli. That's not something that we're going to spend a whole lot of time talking about in this class, but...
So is that saying, like, women can smell when other women...
Yeah.
Then their body's just like, oh, me too.
Yeah, there's a lot more to it than that. There's like a whole discussion we could have on pheromones, right?
Yeah.
Doesn't really fall under the purview of this course, but Kyle can tell you all about it in a few weeks, right? The other class, Kyle, physiological psychology, we're going to spend a whole chapter on...
Depends on how good you teach it.
How good I teach it?
Yeah.
Well, perfect. How good you sort of absorb that might be a little different. That's going to be where the disconnect is, so we'll fix that. Yeah, so a lot of this is based on pheromones. Also, you can initiate, they typically do this in rodents, right, where we would call it an estrous cycle instead of a menstrual cycle. That's kind of a side story, right, it's the same thing. But you can take some mice and like spray urine on them from a male mouse, which is a lot of fun. And you can actually initiate their menstrual cycles that way, which is kind of cool. So I don't know if you like randomly want to go around and start menstrual cycles, you can spray people with male urine. I feel like that's going to get people in trouble, so I wouldn't recommend it, but it's a possibility. I'm sorry, Eli?
You might get an assault charge.
You could. I'm certain there's legislation against pouring urine on people. Is that true?
Only one way to find out.
I think there's more than one way. Like I know a guy who's a lawyer, and I might just call him, or search the West Virginia code of what not to do.
Code of what not to do.
Yeah, I don't know what they call it. But smell. There's a lot of interesting stuff with smell, right? We'll talk about some fun things in a moment. When we're talking about smell, we're really thinking about what are the receptors, right? And so you've got special receptors called the olfactory receptors. I know they got really creative there because it's for olfaction, so we're going to call it olfactory receptors. These guys, guess where they're located? Real shocker here, up your nose. I know, nobody was ready for that. They're up there in the olfactory epithelium, right? It's kind of this like blue space here, and they hang down into obviously like some cavity there, right, where you can get some air. In order for you to smell something, little pieces of whatever it is you're smelling, it's got to make its way up in there. Okay? So next time you smell a dirty sock, cat, whatever. How many of you have a cat? Yeah. Every time you, that's where it is. Cat litter and feces and urine and that other stuff they squirt out sometimes, right? Whatever, that unnamed substance of severe odor. All of that, little pieces of it have to come up in there. Hot dogs, who loves the smell of hot dogs? Nobody's going to own up to that, but yeah, that's the face a lot of people make, right? But little pieces of that has to, these little volatile molecules have to get into your olfactory receptors, right? They have to get in there, and they have to stimulate those receptors so that you can go, yes, I just smelled my cat. So I think that would make me want to keep my domicile clean, right? Okay, because all of those fun things that you're smelling are just right up in there. Socks, onions. How many of you leave like rotted onions around? That's a thing. I have a poor opinion of your living conditions. I just assume they're just like rotted, moldy food and like rabid animals running through.
Well, you know me.
I know, right?
Don't even have a door on it. It's just open to whoever.
You got one of those beaded curtains.
Yeah.
Oh my
Oh my
god.
god.
How many of you have a beaded curtain at home? Nobody's going to raise their hand on that. I know half of you do.
What year do you think it is?
It's not important what year I think it is. It's what year you think it is. They sell these things, I know.
What year do you think we think it is?
I don't know, Alex.
And now it's our turn to guess what year you think we think it is. So we have to guess it.
Do you think?
Yes. I mean, maybe.
Yeah, we'll find out in a couple weeks. What do we need to know about olfactory receptors? Again, they're on the roof of the nasal cavity. If you're a human, assuming most of you are, you got about 6 million of those. That's pretty awesome. They stick out on these little hairs that come from the receptor cells. That's where we're going to pick up these little molecules. What's sort of interesting about this is you have about 1,000 different types of receptors, and all of these are coded by different genes, right? So you've got different genes, they're going to code these different types of receptors, and we're going to code 1,000 different types of receptors. And that sounds pretty amazing, right? What's even more amazing is you can identify about 10,000 different odors, right? Which is pretty cool. So out of those 1,000 different receptor types, you can identify 10,000 different odors. Now you're going to think, well, how am I doing that? Why don't I have 10,000 receptors, one for each odor, right? But we're going to talk about something where we're going to think about the combination of these receptors, combinatorial processing, right? It's going to be pretty exciting. So just hang in there. That's going to be a fun thing we're going to revisit here in just a few minutes. Coming up from the receptor sites, we go into mitral cells. You don't have to answer either of these questions, but is anybody Catholic or plays chess? I got some head nods on both. I don't know which is which. That's fine. Why would I ask this? It's because it's the mitre hat, right, that a bishop wears, and that's why this is important. So if you're Catholic, you get bishops. If you're, Bobby Fischer, you also get bishops. Either way you go. And they kind of have and there's a way you can fold napkins too, right? There's the mitre fold for napkins. Anybody into fancy napkin folding?
You know me.
I know, right? We're just really, you guys have interesting hobbies. So you have these mitral cells. They receive information from about 200 cells. They're all going to be the same type, right? So when you get to the mitral cells, all of those are going to be receiving information from the same type of receptor. So this is kind of an interesting process where we think about something called convergence, right? So convergence means we're taking, in this case, 6 million receptors, right? And we're funneling that down to about 50,000 mitral cells. And you might think, well, 50,000 is still a big number, right? But it's not nearly as big as 6 million, right? So there's like a bunch of extra zeros there for 6 million that you don't get for 50,000. So we're really, really sort of narrowing that down, right? We're converging all of those inputs into one thing, which is pretty cool, but still keeping it separate by receptor type. All right, so what's fun stuff here? We've got our olfactory receptors. This info dumps down into something called the olfactory bulb, right? It's a kind of a bulb at the end of the olfactory nerve. So there we go, so olfactory bulb. From there we go a couple places. One, we go to what's called primary olfactory cortex, right? And then we go a few other places, which is kind of a little bit of an odd setup with the olfactory system, right? This is an exception to the normal sensory apparatus, thalamus, cortex. That's the story I've tried to sell you last week, right? That's the story that is typical for most senses, but it's not the story for olfaction. We can think about why in a few moments. So the other path that we would take is we would go into the amygdala, and from the amygdala we go a few places, including the hippocampus, right? So often you can smell things that you have an emotional response to, right? I mean, people hear that a lot, right? This is a lot more pronounced if you're a rodent, right? So if you can imagine you're a rat for a few moments, I want everybody to just kind of go through their improv training and just imagine they're a rat, like start grooming yourself and, you know, pulling on your whiskers or whatever, right? And just kind of have that experience. And then all of a sudden, imagine like a big cloud of cat urine comes through. That sounds thrilling. It's definitely going to get you excited, right? And sort of upset. And the reason for that is, you know, what typically comes with cat urine?
A cat.
Yeah, a cat, right? And so cats will eat rats, so watch out for that, right? If you guys ever are a rat, cats will get you. So, definitely makes a lot of sense to make that connection between what you smell and the amygdala, right? The other thing is, if you think about humans, how fast is olfaction, right? I mean, it's a little slower, right? I mean, when you see something, you know exactly where it is, you know exactly what's going on, you can identify that quickly, right? If you think about the olfactory cortex, if you smell something, how long does that take you to identify what that is? How long does it take you to locate what that is? It takes some time, right? Whereas vision and audition, you know, we're pretty quick on those things. And some of that's going to be related to the wiring here, because we've got a few extra steps and we're going to some odd places first. It's going to take us just a little bit longer to make that identification. But if we can get that info over to the amygdala pretty quickly, and the amygdala can go ahead and go like, hey, this sort of smells like something I should be afraid of. Let me go ahead and work on getting amped up. If I don't need to, well, I can calm down later. But if I need to be up there, then I can get away from whatever this threat is. It's kind of a cool system. The other sort of interesting thing about the olfactory system is it's probably the, kind of depends on how you want to think about this. Olfaction and taste are probably the two oldest sensory systems, right, that developed. The reason for that is, you know, life started out in this kind of like soupy, watery mix, right? How many of you have ever been underwater? Great. What are some things that you can't do really well underwater? Breathe.
See.
Well, you can't smell because your smell is connected to your lungs. But you can't hear and you can't see, right? What you can do, though, are detect chemicals that are diluted in that water, right? So you could
You can hear underwater.
You can, but not as well as you can above water, right? And you can also taste. So the next time you go to a public pool, open your mouth and take a big
Bite.
bite. Yeah, sure, because you don't know what's floating in there.
Take a bite out of someone's arm.
Yeah, this is why I don't get in public pools. It's like taking a bath with someone.
A lot of someone's.
And there's a very short list of people that I'd like to take a bath with. None of them are at the public pool. I assure you, none of them are there. So think about that the next time you're at the public pool and you're just, you know, flailing around, snorting up water. But because life started in sort of this, you know, diluted kind of situation where chemicals would be floating around freely, chemical senses were the first sort of sensory systems to develop, and then later they developed into other systems. So that's why with olfaction and gustation, which we'll talk about in a few moments, the wiring diagram is a little bit different. It's a little bit older system, right, that we've just kind of latched onto early in the evolutionary process. Didn't really bother to change it. Instead of changing that system, we just said, hey, how about eyes? That probably will work if we're like not going to be underwater all the time, so let's just do that. And it's worked out fairly well. All right, Again, we already go to the amygdala, and we've talked about that emotional connection. Not a big deal. Hey, this is kind of cool. This was a study back in the 80s, and this is still fairly true. There are few sex differences when it comes to sensory capabilities. There are a few, like 2, and we'll kind of talk about those as we need to. The first one we're going to talk about is with odor identification. And as we said, humans can identify up to about 10,000 different odors. However, females tend to do just a little bit better on odor identification, which is kind of interesting, right? So we're slightly better at identifying odors such as ammonia, brute, I don't know why that's on the list, sherry, actually those are all for men, but when you get over to women, interestingly, my favorite odor is Band-Aid. That's that. Right, everybody laughs because you know Band-Aid has a certain odor to it, right? You can identify that. So most of these is just kind of a random cat food. Women are better at identifying cat food smells than men. I don't know. I don't know why. That's just interesting. So women tend to be slightly better at men on odor identification. If you were to give men and women a test of this, you would find that the women would be more accurate in their identifications. And there's some interesting sort of theories about why this might be the case, right? And it sort of goes back to our hunter-gatherer days and sort of some interesting biological differences, right? So if you think about hunter-gatherer days, right? I don't know if, how many of you have ever been in a men's locker room? Best smelling place ever, right, Chad? Okay. I know it's Mason, but I just wanted to toss that out there.
Oh, I was talking.
Yeah, I was actually,
they're great. It's lovely.
they're great. They're lovely. If you've not been in a men's locker room, I mean, I reckon you go in. I suppose you go in just for your own sort of.
Experience.
Yeah, right. Olfactory education. That's the phrasing I'd like to use, right? Olfactory education. Men's locker rooms, they stink, right? You get a bunch of men together, they have a funk, right? It's a true story. This is, yeah, right? It's not pleasant, okay? And if you think about these sort of hunting parties, and you've got like a bunch of these men together, this was also in like the pre-Old Spice days, when there weren't showers and soap and all these other sort of nice fun things to get rid of those odors, right? And you think, well, let's just put a bunch of guys together to hunt down a woolly mammoth. That sounds brilliant. I bet like the first guy was like, man, you guys really stink. And so it's really kind of important to not be as maybe sensitive to those olfactory sort of issues if you're a male. Thinking about it from the female perspective, it might make sense to be able to identify foods that have spoiled, right? Or other sort of noxious chemicals that you might be ingesting, right? Or might be exposed to. Particularly important if you've ever tried to make a half copy of yourself. If you've not, I recommend you don't. You're pretty awesome, I guess, but a half copy of you may not be. Keep that in mind. If you don't believe me, think about your parents, right? So if you're trying to like grow another person inside of you, it's sort of important to go, maybe I shouldn't ingest that, right? Because then it's like a 2-for-1 bang, right? Not only are you going to do something to you, but maybe that thing that falls out of you in a few months, you're going to do something weird to it.
Oh my gosh.
You're talking to
Cool.
That's great.
When I talk to kids, what?
I said when you talk of children.
Oh, I don't
Great.
talk to kids very much.
It's I've
got a
very
nephew
sweet.
that's
I
11.
can tell.
Yeah.
You're very passionate about
The
it.
best part is, it's not all inaccurate at all.
No, it's not.
That's just one way
Well,
to describe
well,
it.
one part of it was the fallout
Yeah.
part. 'Cause they don't fall out. You got to put a lot of effort into that. Trust me. I saw the video. It's too much work for another person. And then you got to take care of them for a while because they're useless for at least 30 years.
Oh my gosh.
Right? How many of you are still relying on your parents? Nobody. Kyle, thanks for being honest.
I got you.
Yeah, right. Because everybody else should have raised their
Well,
hands.
define rely. Be left and right limits here.
Yeah, I still think most of you, if you're not like regularly asking your parents, assuming your parents are like normal people, for guidance, you probably should, right? Because they've made mistakes. You should just dodge those.
I'm just trying to dodge everything they did, so.
Yeah, right.
Living by example here.
Yeah. My dad tells me there's 2 ways to learn things: the hard way and the easy way. I think he lied about the easy way. The hard way is a definite option. You guys can pick that one. which you do a lot. All right, so, hey, remember that time I told you you can identify like 10,000 different things, but you only have 1,000 receptors for that? And you guys have been wondering, like, how does that happen, right? Chandler, you've been sitting there like, I have no idea, right? I'm just like, yeah, I know, right? It's just amazing. How we do this is we really just sort of take different receptors, and they each get activated by a different substance. And some of those substances activate more than one receptor, right? And so it's an interesting combination. It's that combination that's important, right? How many of you have a bicycle lock, one of those barrel bicycle locks, you know, that's got like the 4 digits, 0 through 9, and you spin it, right? You guys have seen those, right? Skylar, and you spin it, you pull it. So you've got 4 digits, or you've got 4 places, you've got 9 digits. That'll actually make almost 10,000 different combinations, right? So anybody have a nice bicycle, they want to share their combination and where they left it? No, nobody's going to do that. So you can spin that around, right? It's the same idea here. You've got 10,000 different receptors, or 1,000 different receptors. We can activate those in different combinations to come up to those 10,000 different odors, right? And so here, I think these are 4 odors everyone's familiar with: cheese, orange, wood, and cognac. It's an odd combination. So there you go. Gives it hanging out in some weird places. So if you take a look at this and you think, oh, well, I'm smelling some wood. It activates this receptor type and these 2 other receptor types as well, so that's cool. Well, then I'm smelling an orange. Well, orange doesn't activate that receptor, but it activates a different receptor here. We've got a little overlap, right? And then sort of 2 other receptor types that are going to be activated. So it's different combinations, right, that are going to tell you this is orange, this is wood, this is cheese, this is Band-Aid, this is cat food, whatever it is, right? We've got 1,000 different receptors that we could activate. We'll activate different combinations of those for each of those 10,000 different odors. There's going to be some overlap, but there has to be something that's a little bit different as well. Okay? Are there questions about that? Because this is actually sort of an important concept. All right, we'll move on. Did you have something? You look like you had a question. You sure? Okay. We call this population coding, right? So it's not the activity of one receptor that's important, it's the activity of a whole group of receptors that's important. It's going to tell us what's going on. Okay. It's that pattern. And this is how we know. So if you've got 10 receptors and 3 of them are activated by cheese, and 2 of those and 1 different one is activated by orange, then you know, hey, that's a different pattern of activation, even though there's some overlap. We know it's a different substance. And that gives us a lot of flexibility. That way we don't need 10,000 different genes for 10,000 different receptors, right? We only need 1,000 receptors and 1,000 genes to code for those. It's a little bit efficient. Hey, who loves odor adaptation? Anybody who's ever smelled something bad, right? How many of you don't have a cat? Yeah, Olivia, and you visited someone who does. Yeah, and you go in and you're like, whoa, man, are you breeding skunks?
Or, my brother's room.
Yeah, right?
It's freaking horrendous.
So you go in there and it's like, oh, jeez, this is awful, right? And you go, okay, well, what am I going to do? You've got 2 options. You can leave or you can just stay there for a few minutes. And then once you stay for a few minutes, you're like, well, this really isn't so bad. Someone must have done something, like giving their cat away. That's not what has happened, right? You can give their cat away. What's happened is you have adapted to that, right?
Is this like that idea of like those commercials where they've gone nose blind to certain odors?
Yeah, I don't know if I would, nose blind, you can use that phrase. Febreze will come and sue you, but you're welcome to do so. But yes, it's that idea, right? Once you've adapted to an odor. Now, what's interesting about this is, once you leave and come back, you would have to readapt to that odor, right? Because you would be like exposed to it again, right? It's not, adaptation doesn't last forever, right? You kind of have to, you get used to it again and again, but it's not a big deal. Has anybody ever, this is going to be a weird question, how many of you have ever dealt with dead bodies? Okay, a couple people. That's interesting. So if you've ever been in like a cadaver lab, right? Or if you worked at a funeral home, you know, that's as far as we're going to go. I'm not going to ask what else you were doing as to why you were dealing with dead bodies. But there's like an odor, and you get used to that after a little while, and you're like, okay, I'm fine. And you just move on, right? So it's adaptation. Anybody interested in working with dead bodies? Okay, no. Had a project, never mind. Trying to recruit. So, you'll, over a brief period of time, kind of lose sensitivity, right? You remove that stimulus, and pow, it jumps right back up. And, you know, you have that same kind of initial response to it later. So there you go. You can get a drop of about 30% after about 12 minutes, right? This is why people can't smell their own house or their own body odor, right? Have you ever been away from yourself for more than 12 minutes?
Yes.
Not usually.
Not usually. It's difficult to do, right? You have to be really fast. It can happen. So, you know, you're not going to smell yourself, right? You're going to go up, I smell nothing, right? Because you've adapted to that. You're always constantly around yourself. So there you go. There are also some things called cross-adaptation, right? So if you smell one thing and then you smell something kind of similar to that, then you will also reduce kind of the perceived intensity of that other sort of, you know, object as well, right? So last week we had some lemons, or lemon juice, and limes, right? I would say lemons and limes have a similar odor, right? You know, if you had a gerbil and a brat, I'd say that smells the same. You know, you can kind of cross-adapt to some of these things. But it's not going to be as powerful. Hey, here's kind of an interesting thing. This is true odor blindness for you, Alan. It's called anosmia. It's when you actually don't have the ability to detect an odor, right? This can happen for certain odors, right? Cyanide, that's one you do want to be able to smell if you can, right? You never know.
Carbon monoxide.
Yeah, right. That's one. Butyl mercaptan is kind of an interesting thing too. Anybody have gas, heat, or stove at their house? Yeah. So they will stick this butyl mercaptan in your gas so that you can smell it, right? So that's why if you've ever like smelled gas or a gas leak, you're like, oh man, that's awful. But if there's ever a gas leak and you're not able to smell that, well then you're probably nose blind to it. 1 in about 1,000 people will be nose or odor blind to that, right? So that's kind of interesting. Some of this is genetic, right, which is kind of interesting. Sort of another interesting story about anosmia is that it's actually like losing your ability to smell is one of the first signs of dementia, right? It's one of the first systems to go out. And so you should really, you know, give yourself like, you know, an odor test every day, right? So you should have like these small vials and go, okay, it's good. And then you got to do like your vision test, you know, so you're not losing your sight. You do a quick hearing test, and then you're ready for the day. Actually, Parkinson's disease, Josh. Problems in the olfactory bulb kind of happen early. Alzheimer's disease, all of these things, olfactory bulb kind of kicks out first. People don't realize it because we're not, again, constantly relying on odor identification, right? And if things get bad and it's during cold and flu season and you've got some sinus trouble, well, you'll never know. Now, you have a question, or sure? Okay, let's have it.
I'm in Dr. Atkins' adult development, and she was talking about as people get older, naturally they'll like lose taste. Is that because of the olfactory bulb just diminishing?
There are, and we'll talk in this class about when we get to the sort of the end, individual differences, age-related differences. There are a ton of things that go on. Short answer is, as you get older, things die, and you're going to lose cells in all parts of this process, not just like out in the sensory apparatus, but also in the cortex and the other processing areas. But yeah, that's a normal age-related process. So if you do start to kind of lose your sensory capabilities as you get a little older, that's normal. It's kind of that drop-off. You know, it's how fast do you have that drop-off, right? That's what's really important.
Okay.
No, that was a good question.
Can I ask you a question?
Go ahead.
What about people who like gag or almost throw up when they smell something that has like made them sick before?
They're overly dramatic. You should not be friends with those people.
Okay.
Just move on.
You know, not saying
Yeah.
not for me personally, but
Right.
you know, people who smell certain kinds of alcohol or.
Yeah, so taste, there's a thing called conditioned taste aversion, right? Which is super powerful. And that's a great segue into the next slide, which is about taste. So great job.
Thanks.
So conditioned taste aversion is this idea that when you taste something or you eat something, ingest something, and it makes you sick, you're not going to come back to that, right? And that's awesome. It's one of the few examples, if not perhaps maybe the only example, of what we call single trial learning, right? I've not eaten a hoagie sandwich since the 3rd grade. I ate one once and it made me sick, and that was it, right? So I've not gone back there to get hoagies anywhere, right? I'll eat a sandwich on a circular or a square bun, but not the oblong bun. It just doesn't work. I don't know what it is.
So the shape of the bun
The
is what
shape
did
of the
it
bun
for you?
is the shape of the bun, guaranteed. Is that's what made me sick. And how many of you have a place like here in town where you won't eat because you ate there once and you got sick? Yep, everybody does.
Towers.
Huh?
I mean, I really got sick from them, not just, food was just gross.
Yeah.
Or the restaurant itself was gross and the food was mediocre.
We're gonna get you a restaurant review column in the Herald Dispatch. At the table with Alex Spiegelberg. It's gonna be a great column. It's not bad, right?
Not bad.
Think about it. They might even put you on the local radio station, right? Get a little content for WMUL.
Make a
And then everybody will
podcast.
find out that I'm always on
You
the
can make a
81
podcast.
thing.
You can make a podcast. Yeah, you can make a podcast. There
Then he
you go.
moves on to TED Talks.
TED Talks. Yeah, then CNN hires you for their, like, food analyst. I don't know. So you got a whole
And then I
career.
accidentally do something stupid on air and get forgotten and thrown out immediately.
That's going to happen before you get to CNN. Trust me. We know that. So, conditioned taste aversion, it happens. It's pretty awesome. Because taste is really important for, one, edible and nutritious foods, but also avoiding poisons, right? And if something makes you sick, it's probably poisonous. You shouldn't eat it again, right? I don't know, I had this great video clip of a blue jay eating a butterfly and then vomiting the butterfly, which is kind of interesting. And so it happens all the time, not just in humans, other species, right? So Charlie was just like, eating this monarch butterfly, right? And it's like, oh, that's just nasty. And then, because, they're kind of poisonous to birds, right? Which is awesome. So if you don't want to get eaten by a bird, you should be poisonous to that bird. That's, it's a brilliant evolutionary trick there. And then what another sort of butterfly might do is like, I'm just going to make myself orange. I'm not going to bother with the poison thing. That's a lot of effort. Make myself orange, and I know that birds won't eat me because they'll think orange butterflies, I get sick, right? So you'll avoid that. So conditioned taste aversion, it's a great thing. Just helps you, again, avoid poisons, which poisons will lead to things like death. Or worse.
That's a big one.
Yeah, that's the big one. I kind of like to start at death, right? Because if it's, but you could have like a lifetime of torturous stomach pain, right? And that would be very fun. So there you go. Taste is great. It is sort of universal in the animal kingdom, right? Almost everything has some form of taste, some way that it's going to ingest some molecules, right? To get to figure out what that is and identify that. You got a bunch of taste receptors. Here's a real shocker. They're on the tongue.
No way.
I know, right? They're also in the mouth and the throat. So there you go. That's a new one. You have taste buds, which is actually somewhere around 50 to 150 receptors, right? They're in there. Taste buds don't live very long. They live about 10 days, and then they get replenished. Okay, not a big deal. They collect that information and send them down the sensory nerve fibers. All right. Now you have sensory nerve fibers for different sort of tastes, right? You've got like sugar, salt, acidic, and then this is quinine's bitter, right? And so you can see that each cell or each fiber gets a little bit excited by every sort of class of taste, right? And there's one more class that we'll talk about in a few moments, but these four we'll think about here. But it's how excited do they get about a particular substance, right? That's really going to tell us. So even though this guy here gets a little bit excited about things that are salty, he gets super excited about things that are acidic. So we know that it's going to be sensitive to acids.
Would that relate to population coding?
Yes, absolutely, right? Because some of these other cells and those taste receptors are going to be picking up these other molecules, but it's, again, that sort of package of activation. This is particularly important because you can identify a number of foods, right? You don't just go, well, that's salty. You go, no, that's salty French fries, or that's salty something else, right? You can tell. Alex?
What exactly is quinine?
It's just a bitter. You've never had quinine? Can't believe you're.
I mean, if I did, I didn't know.
I mean.
We can treat malaria.
Yeah. Yeah, it's a drug. That's really good.
Quinine RX.
Yeah.
That explains it. I've never had a parasite. No wonder I've never tasted it.
Yeah. Yeah, I mean, I don't know that you'll get malaria here, but, you might pick up a parasite soon enough. Just keep eating at that favorite restaurant of yours. Wherever that is. All right, so when we taste things, what happens? A lot of stuff, right? These looks complicated, but this is all taste bud, right? We're going to send all that information into somewhere in the brainstem. Don't worry about that too much. Hey, look at this. We also go to the amygdala, right? We talked about that guy before. That's kind of the center for emotional processing, right? We'll go to the hypothalamus. Hypothalamus is like a hormone guy, so it's really going to control hormones. Why is this important? Well, when you eat different things, you can cause different hormones to be released. That can help you go, well, I'm not hungry anymore, or I'm still hungry, or I need to do something else. And we're also going to send information to the thalamus and then on up to the cortex there. So there are taste dimensions, right? So previously folks had talked about sort of 4 taste dimensions: salty, sweet, sour, and bitter. What's kind of interesting about this is these receptors are looking for specific molecules, right? Specific chemicals. So if we're a receptor sensitive to salty, what kind of molecule do you think we might be looking for there? It's tricky.
Salt.
Yeah, specifically sodium, right? Okay. What about if it's sweet?
Sugar.
Specifically glucose, right? So we have glucose there. If it's sour, that's a little trickier. Huh? Acid. If we're looking for the specific molecule, it's going to be hydrogen ions, or what we call protons, right? Because when you're measuring, if something's acidic, you're going to measure that concentration of hydrogen ions. The higher the concentration of these hydrogen ions, the more acidic it is. For bitter, these are typically like plant alkaloids, right? So things like that. Finally, we have this other taste called umami. Right? This was originally described in Japan, and it sort of means savory or yummy, right? Often associated with monosodium glutamate, or MSG, right? Not anything to get too worked up about. I mean, be concerned about the sodium component of MSG, right? Because that'll give you high blood pressure and a heart attack. The glutamate part is just an amino acid, and that's really what these receptors are latching onto, is that glutamate, right? And they're identifying that particular component. So it's a pretty standard food additive. Hey, who remembers that time I made you eat lemons or limes and drink a bunch of water, and then I tried to give you diabetes by forcing sugar down your throat? That was a lot of fun, right? So what we were looking at there was cross-adaptation to taste. Typically, if you adapt to a sour taste, right, or if you adapt to a sweet taste, it'll affect the taste of things later. So usually if you will have a pretty intense sour taste in your mouth for a while, and then you drink plain water, it'll actually taste a little bit sweet. Okay. This opposite will happen if you have something really sweet, and then you drink some water, often it'll taste sour. All right, we could think about theories of taste coding if we wanted. Let's see if we're moving along fairly well with the time. How many slides do we have left? Oh, only 3 slides, and then we get to move on to body senses, so that's awesome. Yep, Mason?
Just to go back to those different types of taste.
Mhm.
When you lick a 9-volt battery, is that bitter or sour?
It's not murder.
Well, because
Because, so he's going to give us an explanation on why you do that.
I'm not saying you need to lick batteries. I'm just saying it's what you do as a child.
Yeah, there are
So
some.
when you lick a power outlet,
Vastly.
No, not a power outlet, specifically a 9-volt battery.
Yeah, so I've heard of people doing that, but I've never seen one make it this far in life.
what do you lick?
The receptor. You just stick it on your tongue. You don't actually lick it like a lollipop.
Yeah, I think that's really just that electrical signal hijacking your entire sort of.
So you're not really
Conduction.
tasting anything.
I don't think so. I mean, I think if you were to lick somewhere else on the battery, it might be like a bitter taste, like metal sometimes has a bitter taste. But I don't know. I mean, go home and try it if you want, at your own risk. It's an interesting question.
I'm alive. It's
Yeah,
okay.
yeah. I guess that's all that matters, right?
Yeah.
I should mention there are a couple of other actual taste dimensions that we're not thinking about here. One is spicy, which is sort of a separate process, right? So the intensity of something that's spicy. There's also some evidence that there might be like a bread or carbohydrate receptor. Yeah, this is pretty new actually, new research, and so there are some folks looking at receptors and DNA behind that, and they think that at least in some populations there seems to be a receptor for like carb bread kind of things. It's kind of interesting.
Why would they need that?
Huh?
Why would they need that?
Why would you need that? Well, you need carbohydrates. I mean, that's a standard unit. So any of these things that you would have a taste receptor for, you either need it to ingest it or you need to avoid ingesting it, right? So for like bitter, that's something you would need to avoid ingesting, right? Because bitter things tend to be poisons, right? And so that's what we're thinking about there. But you definitely need hydrogen and acidic things, particularly if you remember that story about limeys. Particularly if you don't want to fall apart, you're going to need certain kinds of acid there from limes. Sweet things, glucose, you definitely need glucose, right? Your brain runs exclusively on glucose. Okay, so you can feel a little bit better about all those brownies that you eat. No? You can make glucose out of other things.
Right, so if there is a carbohydrate receptor and all carbs turn into glucose, why is it just a version that's sweet?
Really, there's only been a conversation about this for the last year. So it's fresh. So it's kind of like we don't really know. There's some evidence that folks are, like again, in some populations, that folks that might have a genetic variant for a carb receptor. So it's a very new, it's a very, sort of interesting thing, actually. So.
Could it be that these people just eat so many that it develops?
Probably Italian.
Could it be that they're just crazy?
No.
I was thinking that like maybe if
So
they
they're
live
fine.
somewhere you can't get
Exactly.
carbs easily, maybe they'd like.
Yeah,
need to
I
do
mean, all of
something.
these things, I mean, and some more so than others, are reasonable explanations. But some of them are less reasonable. Alex, you can pick which one that was.
Everybody else's was the least reasonable. They're all insane.
Right. So, I don't know. I mean, it's sort of interesting, right? I mean that it's. I don't know. We'll find out or we won't. There's also like a whole group of people who, when they eat cilantro, it tastes like soap, apparently.
Yes.
Yes.
Yeah.
Yeah, it's very soapy.
I don't know what you weirdos are doing. That's
And
actually.
they judge me for licking a battery.
I still think that's worse, man.
Licking the
You
battery.
can't help what
Yeah.
cilantro tastes like.
Yeah.
You have to actively lick a battery.
At least you're
to find out.
supposed to eat cilantro. Like, at least it's edible.
Yeah.
I guess. So that's related to a genetic variant in your taste receptors as well, so which is kind of cool. Right, Marissa?
Yes.
Are you in that group? Yeah. Well, do whatever you need to with that.
I just don't eat it.
Yeah. Just, get some Irish Spring or something.
I have a question.
Okay.
So like,
It's not soap related, is it?
no.
Great.
But like, sometimes, like, I can't tell which direction it is, but sometimes I like taste the smell of something or smell the taste of something.
Yeah,
It's
so
really
there-
weird. I don't understand.
There's a definite, and we'll talk about that in a few moments. So no worries. What do you need to know about theories of taste coding? Not really. Not a big deal here. Basically, we've got these sensory fibers. They'll respond best just to one taste modality, right? So one taste. That's really all you need to take out of that. Conditioned taste aversion, we've already talked about this. Works in about every single species, including reptiles and fish. So that's pretty cool. Right? This is really important if you know someone who's undergoing chemotherapy or radiotherapy, right? Because that definitely has a massive effect on like how much you vomit and how you if you feel sick. So if you're ever in one of these situations, don't eat your favorite foods at that time, right? Eat things you already hate, because it's like, oh geez, I don't know, tuna. I hate tuna, so I'm gonna eat tuna while I'm undergoing chemotherapy because it's not gonna make a difference to my life later. Don't eat like chocolate brownies. If you love chocolate brownies, don't eat them while you're going through chemotherapy because you'll vomit and then you'll have conditioned taste aversion, and then you've lost brownies, right?
Do you actually lose taste buds? Like, you know how someone will say, like, if you burn your mouth, then you just lose those
Well, you
taste
can
buds?
obviously cause damage. I'm like licking batteries.
I'm talking about like hot coffee.
You can cause physical damage
Yeah.
that could cause you problems. I wouldn't imagine if you're just drinking hot coffee on a regular basis, you'd really cause a problem. The biggest problem with drinking hot beverages is you can develop esophageal cancer. That includes like hot teas, or if you guys drink hot orange juices.
Ew.
Why would you drink
Hot orange
Oh,
juice.
That's
Trying to start a trend. Josh, did you have a question?
What about, okay, so I have like a bunch of older people in my life who say,
Are you in their wills?
huh?
Are you in their wills?
I mean, I don't know.
Well, that's the first thing. If you've got a bunch of older people in your life, so fix that.
Okay,
Get in their
I'll
wills,
figure
and
that
then,
out.
then we'll move forward.
They always say like, oh, I've been smoking for such and such years, I can't taste anything anymore.
Yeah.
Or I
That's true.
went through all these cancer treatments, so now everything tastes like nothing.
Yeah, I mean, all of that causes, so when you go through chemotherapy, right, so here's like a little side story I didn't really want to tell, but I'll tell it anyway. The reason you have problems with your stomach, the reason your hair falls out, is because it specifically targets cells that are dividing, right, like tumor cells. And it's going to kill cells that are constantly dividing and growing and being replaced, right? And so how often does your hair continues to grow all the time? Your stomach lining is constant, right? There's always cells in there. Remember that time I told you every 10 days you're getting new taste receptors? It's going to be targeting those cells as well. Okay? So yeah, makes a lot of sense.
I understand.
Yeah, it it really does make a lot of sense, right? It all kind of fits together. It's kind of a cool thing.
Full circle.
Yeah, I told you, this class is a carousel. We're coming right. Didn't know it was all related. I think what's important about this discussion is a lot of things, but I think one thing is how important sensory systems are to understanding human behavior, right? And what we do. And day to day. We don't often think about our sensory systems and the influence of those sensory systems on our behavior and what the limits of that sensory system is and how that influences what you can and cannot do. You don't think about it. We just, when we talk later about the visual system, there are some species that see ultraviolet light. What's ultraviolet light to us? Nothing, right? I mean, it doesn't change our sensory experience or our perceptual experience, right? Because we're not sensitive to that in any meaningful way. And it's really hard to imagine what that would be like to see ultraviolet light and how that might influence your behavior, right? Because we just think, well, visible light, we can see everything that there is. But we can't. We see a sliver. We think about taste, and we think, well, we can taste everything. We can taste a sliver, right? There's some things we're going to be tasting, it's not, it's going to be different. It's not going to do anything for us.
Like specifically for light, it's the mantis shrimp, I think.
Yeah.
It's just some random aquatic animal. I think you can see like 12 more colors than we can or something.
Yeah, it's quite possible. There.
And the video that said that was like, imagine a color that you can't even imagine. Now do that 12 times.
Yeah, unfortunately, I don't know the mantis shrimp personally, but.
You spray this thing, they suck. They'll punch you.
They're really cool.
Well, maybe we can bring some in for class one day.
No, don't do that.
No. Are they very big and tasty?
No, they're like this big.
Oh, really?
Yeah.
Huh.
They're cool because of how dangerous they are.
They're not cool because of how dangerous they are.
Field trip.
They can create underwater shockwaves.
Yeah.
Just with their little.
I know about that. Yeah. Pretty impressive. So who wants to talk about flavor? So this is where we're talking about like smell and taste, right, working together, okay, to sort of create flavor. But there's even more to it than that. There's even like temperature, touch, sight, sound, pain, right? All of these are contributors to flavor. Visual color, this is kind of an interesting one. Anybody know why they put artificial colors in most everything you eat? To trick you into thinking it tastes more powerful than it does, right? What's cheaper than strawberries? Strawberry color, right? Okay, so if you're going to drink a beverage or eat some yogurt that's strawberry flavored, instead of putting in actual strawberries, you can just put in some red food coloring. And so if you were to drink a beverage that was more powerfully red than another beverage, you would rate that more powerfully red beverage as more strongly flavored, even if in fact it wasn't. So there you go. This is also why, I don't know if any of you drink I see these like vegetable fruit juice blends, right? If you ever notice, they put those wrappers on there that cover the entire thing so you can't see what it looks like, right? Because when you pour it out, it looks like that, if you ever cut your grass when it's wet, right? And you get that kind of mass of wet grass. That looks like that's what you're pouring out of that cup or out of that bottle, right? You don't want to see that at the store because you'll never buy it, right? I promise you. Look at that. Like, I'm buying that stuff. Okay, so they try to trick you.
Where does that stem from? Like, how does that benefit us as humans?
Yeah, well, I mean, it benefits the people who are like trying to sell you things.
Well, yeah.
Honestly,
How do we develop that?
yeah, so if you think about this, I mean, in nature, it's going to be, like if I was just like out and about, like doing my business, and I was going to try to, like, I don't know, get some strawberries, right? So if you think about strawberries, like themselves, strawberry flavored things, those that are more brightly colored red, guess what? They're riper and they're more flavorful, right? And they taste better for you when you eat those. If you eat the ones that are, they've got the little white on the end, right? Sometimes you get those aren't any good, right? You don't want to eat those. And so I think, if we were in our natural environment, it makes perfect sense, right? It helps us get the more flavorful foods that are ripe, that are not going to cause us like digestive problems. They're going to be, more healthful for us, and that's going to be a better situation. What I think has happened over the years, I don't know that people have always consciously thought about this, although I think some people have, because there's some nefarious folks out there that are trying to sell you things to put in your body, right? And so they'll take advantage of these things. And it happens all the time. This is another thing, why are there so many health issues related to food intake, right? So if you start to think about what are some of the biggest problems, and not just in the state of West Virginia, but across the nation, obesity and some of the related, diabetes, high blood pressure, right? All of these things, a lot of those are dietary, at least diet contributes heavily to those things, right? Some of it's genetic and it's harder to avoid, right? but it's because foods, right? I mean, when the sweetest thing you could get your hands on was an apple, nobody was gonna get diabetes, right? You're just eating
I mean,
apples.
you might,
You-
if you eat
you-
100 apples a day.
You might, but, I mean, at least you'll be regular.
It's more about calorie
well,
intake.
But that's different than like cheesecake, right? I mean, you get your hands on cheesecake.
Calorically, if you eat McDonald's, but you eat 3,000 calories in McDonald's, whereas if you only eat like an Egg McMuffin, you're not necessarily gonna get obese from that.
You're not, but it's so much easier to get these calorie-dense foods now that are high in salt, high in fat, and high in sugar.
And high fructose corn syrup versus regular glucose is much harder for our body to process, so it's pretty rough on your body.
Josh, you got something on that?
waiting on you.
You're waiting on me? I'll let you handle
You,
it.
Professor.
I'll let you handle it.
No, I'm good.
You sure?
Over here.
Not over here.
You're over there. I think there's some debate on that, right?
Yeah, because sugar is sugar to your body. Doesn't really differentiate between processed sugar and fruit sugar.
Yeah. I mean, I brought you in organic cane sugar just because it was in a convenient bag. Don't think that's any better than just like the syrup, right? We can just drink molasses.
True.
I think the thing with that is it's just cheaper to put in foods.
It is,
I-
and
I think that's
to get it into certain types of foods too, it's easier. So I think it's, yeah. Don't buy into the like, those anti-high fructose corn syrup folks. Although there are some folks, there's an interesting diet out there called the 5-MAP diet. I don't know if, it's kind of a newer thing that Stanford developed a few years ago, but a lot of that is for folks who have, there are folks who have some digestive problems, and they want you to avoid like long-chain polysaccharides, which includes high fructose corn syrups. But you should avoid just about like a lot of other things that fall in that category as well. So, yeah, so you're good, stay on the high fructose corn syrup, you're fine. As much of it as you want,
Fair enough.
within moderation.
Where does fake sugars come into this, like Splenda and all that?
Fake sugars.
Well, no, because like I know that they are processed differently, right? That's why some people are like, if they eat them, they die. PKU. That's a thing, right?
Yeah, I mean, it's a thing beyond discourse.
Okay.
They've got that, where I would go is down 4th Avenue, and there's this place called Huntington's Kitchen. Just walk in there and start screaming about fake sugars, and someone will help you. I promise. They've got a whole team of people down there. Just go, fake sugars are invading my life, and then they'll give you a pamphlet.
Okay.
You'll be fine. So that's the end of taste and smell. So that was something. You guys had some questions. Hey, don't feel bad. I tested an electric fence one time with a blade of grass. It will conduct electricity, just in case you want to know. Just in case you want to know. I was like 7. I was like, hey, I wonder if this works. My grandfather was like, well, there's one way to find out. I'm realizing now there was another way to find out, but, at 7, what are you going to do? So I pulled up a blade of grass, pow, and it worked.
Do you know how strong the pencil is?
Huh?
Do you know how strong the power on the pencil is?
I don't. I don't think it was too bad. I really just felt it in my hands. I remember it felt like somebody broke my finger. Like I was like, wow. Yeah, it was like, that really hurt. And I was like, I'm not going to do that again.
I got shocked by electric fence. I think it was like 2,000 or so. Felt it all the way up to my shoulder.
Oh, wow. There you go. Was it for scientific inquiry or just an accident?
Pure accident.
Pure accident, that's okay.
That's okay.
Accidents happen.
I mean, you're not wrong.
They do, right? That's why we call them accidents.
Yeah.
Otherwise, we would call them no-cidents because they didn't happen.
So when you tested it.
I just made that was good, wasn't it? No one laughed because I don't think anybody got it. It's more of a sight gag than it is a verbal gag. But I think it was good, Caitlin. Thank you. You were thinking about that. All right, let's think about body senses for a little bit. We'll shift gears because we need to get through that. No, don't worry about that picture. It's not important. Hey, but we do need to think about touch, right? So there are a bunch of things we want to think about with touch and proprioception and kinesthesis, right? So proprioception is position of your body. If it's at rest, right? If it's kind of hanging out there. Most of you know where most of your body parts are right now without having to look at them. That's pretty awesome. If you ever get up and move around, you'll know that your body is moving, and you will know where they're going and what they're doing, right? Because you have special sensors for that. Not a big deal. I'm not going to hit you too hard on this. I do want you to think about how we might put, especially for touch, right? Because we're going to embed these things. Sometimes we call them the cutaneous senses, right? Because they're embedded in your skin somewhere, right? And so if you think about what are all the different sensations you can experience with your skin. You've got hot and cold, you've got pain, you've got pressure, you've got vibration, touch, you've got tickle, itch, texture, right? All of these are mediated by different types of receptors, okay? I'm not going to ask you to memorize those types, but think about the structure a little bit where we might locate those. For example, if you want to put something, if you want something that's sensitive to light touch, you're going to want to put that toward the surface of your skin, right? You're not going to want to put that deep because if something is touching you and lightly and you want to feel that, if your receptor is too deep, it's not going to be stimulated. Okay? Deep receptors, though, that's going to be more for pressure and stretch, so for those more, sort of stronger responses. Alex?
Are there any cases where, say the deep receptors, say to a birth defect, end up on the very top of the skin?
I assume that's possible. I don't know anything off the top of my head, but there are folks who are more sensitive to touch than others, and sometimes that can be problematic. Here are the names of those things and where they're located. Again, don't worry about that too much. Just keep that in mind. For the somatosensory pathway, you've got sort of two pathways going up your spinal cord, because you've got to collect all that information, right, starting from the outside. You've got to send that up the spinal cord to your brain. Different information is carried in each of those pathways, right? So spinothalamic is going to do what we call sluggish pain and temperature. The lemniscal is going to carry fast signals from your mechanoreceptors. Okay? You've got a nice little
What was the other one? What was the second pathway?
So you have the spinothalamic and the lemniscal. And here you go. This is your spinothalamic tract. And we call it spinothalamic because it goes from the spinal cord to the thalamus, right? Very, very creative name. Okay. It's like a bus route. How many of you have ever rode the Milton bus? Guess where it takes you? It takes you to Milton. I don't know if there is a Milton bus.
There is.
There is? Okay. I've never had a reason to ride the bus to Milton. Actually, I've never ridden the bus in the city at all. Huh? Don't do it. Thanks for the advice. You also have your lemniscal tract, right? That's going to go up to your brainstem and then to your thalamus. You've also got some things going over here. These are like reflexes and stuff. Don't worry about that too much. Eventually, we get that info up to the primary somatosensory cortex. That's pretty awesome. We really haven't talked about cortical organization with the olfactory system or the gustatory system. There's an important reason why we didn't do that, because they're not organized, right? They're kind of haphazardly arranged in just whatever way works out by random, right? And that's fine, because there's not a logical way to arrange taste, right? Should I put all the bitter cells next to the sour cells, or should I put those next to the... It doesn't make any sense, right? Why should we do it that way? But when we get to the body senses, it does make some sense to have this cortical representation be highly organized, okay? And basically, you've got your primary motor cortex, right, or primary somatosensory cortex, rather. And on that is kind of a shrink-wrapped and distorted version of the human body. And we call that the homunculus, right? So if you ever want to insult someone, just call them a homunculus, right? It means they're like a, little distorted, kind of person. I'll give you all kinds of great insults in this class, but that's one. you have your cortex, it's got 6 layers, right? Lots of stuff is coming in and out of that, not a big deal. What is kind of important here is to think about something called cortical magnification. And we briefly talked about that last week, right? And this is when I said some parts of your body are more important than others. Okay, you guys remember when I said that? Okay. If we're thinking about your body, for example, your hands are much more important than your shoulders, right? What can you do with your shoulders other than keep on a shirt? That's about the extent. You carry a backpack, but you're not doing anything fancy, right? What are things you can do with your hands? You can play the piano, right? You can play Jenga. I'm sure that's what you spend a lot of your time doing. You can do a ton of things with your hands, right? And there's a lot of fine motor movement there with your hands and your fingers and so forth. So they get a much larger representation, a much larger chunk of your brain goes to your hands than what goes to your back, right? Because again, your back's not that important. Other places that are pretty important that get a lot of area of your brain is going to be your lips and your mouth, okay? And that's because vocalizations, right? When you speak or you talk, you really, I don't think people realize how complicated those movements are. This is important for today's laboratory experience when I asked if you had all received a tetanus shot, right? Because we are going to stab people, right, as deeply as possible with a compass or a pencil. No? You're not into that?
I don't want to be stabbed either.
You really don't want to be stabbed?
No.
We're not going to stab people.
Okay. You sure?
Well,
We're gonna poke them?
what?
We're gonna poke people?
Slightly.
It's okay.
Is that okay?
We're not gonna
That's
try
That's
to
better
stab
than being
people.
stabbed.
Poking is slightly better than being stabbed. What if we just touch you with a sharp object? See, I don't even know if poke, that's a little strong, right, Marissa? We're not really going to poke. We're just going to touch you with a sharp object.
Just touch, just a touch?
Just a touch.
Okay.
But we want to think about this idea of the receptive field, right? It's easy to think about with your skin. So if you think about your arm, right, and you think about your forearm, for example, that's represented somewhere in your brain, right? There's a little space up there for your forearm. And that forearm has a bunch of cells, right, that are sending information up to your brain. But each of those cells is only responsible for a small sort of portion of your arm, right? And when you move out of that receptive field, right, the area where it can receive a stimulus, you've moved into another cell's receptive field. So when you move over just a little bit, another cell is going to be responsible for that information, not that original cell. Okay? Now, this is sort of the basic organization of a receptive field. Later, when we talk about vision, we'll talk about these in a bit more detail because they're more complicated. But the basic idea is with a receptive field, there's a part that's going to be excitatory in the center, right? So if we were to poke someone there, not stab, we're going to poke someone there, that would get that cell excited, right? Because we would know we'd be able to localize that. Now, if we wanted to move outside of that area into what we call the surround, so there's the center, there's that part in the middle, and then the surround is out here. If we move out to the surround, and we were to kind of poke someone in the surround of that receptive field, that cell would actually not get excited, it would actually be inhibited, right? And so this gives us a nice sort of idea, Kyle, about the location of that stimulus, right? Because we've got an excitatory part, we've got an inhibitory part. That's of course going to overlap with other cells, right? And so we can put this in here in a way that we know, yes, it's here, or it's not here, right? So that's important, like knowing where you are, but also knowing where you're not, right? And so we kind of have that nice organization. This will make a lot more sense later when we actually do this lab and when we think about the visual system. Receptive fields play a bigger role. But the basic idea is if we activate the center part there, A, right, we get a lot of action potentials. If we activate the surround area that's inhibitory, we don't get a lot. And if we sort of activate the whole thing, we sort of reduce the activity, but we still get just a little bit, right? Because the positive and the negative, they sort of counterbalance. Okay.
Does this have to do with like the nerve in your elbow? Like with that, is that a different thing?
That's a different thing.
Are we talking about that?
No, it's not important. You don't have to worry about it. All right. 2-point acuity. This is what you're going to do in the lab activity later today. So we need to get to. Well, we're getting there, right? We're gonna have plenty of time. So, what we will have you do is, for the lab, we'll give you some instructions later, but basically 2-point acuity is this. How far apart do we have to put 2 points that are gently poking you in order for you to know that's 2 and not confuse it with 1? And so what we're really asking is, how big is that receptive field, right? Because if both points are inside the same receptive field, guess what? You think it's 1. When we get that second point into a separate receptive field, Alina, then we know it's 2, right? And so then so that's pretty cool, right? Now, the interesting thing about this is, receptive fields have different sizes on different parts of your body. And that translates back into that idea of cortical magnification, right? So if I've given a lot of space in my brain to a particular part of your body, those receptive fields are going to be super small, right? Because I can put a bunch of them in there and I need a bunch of cells for that. If I said, well, you're only getting this much brain space for your back, then the receptive fields need to be large because I don't have room to put a lot of cells there in my brain, right? That makes sense? So it's kind of this inverse relationship. And you can figure this out by measuring it with this 2-point acuity. And so here this is measured in millimeters. That's the accepted scientific length measurement. So if you think about places like the tip of your tongue, your index finger, your lips, other parts of your tongue, even the palm of your hand, look at how close those 2 points can be, and you still know it's 2 points, right? They can be as close as just a couple millimeters, and you know, hey, that is 2 separate things that are poking me, right? If you move out to, let's say, your neck, now we're talking about something that's like 50 millimeters apart, right? So that's a bigger distance, and you still think, well, that's 1, right? So it can be this far apart, and you're like, well, you're just poking me with one pencil, not 2. It's pretty impressive. So receptive field sizes are different in different locations. This is important. Your receptive fields don't need to be large on the back of your neck. You're not really doing anything that requires a lot of sensory input from there. When you're thinking about your hands and the way you use your hands and your fingertips to interact with the world, right? Having a lot of receptive fields that are super small makes a lot of sense, right? So if you could think about like Braille. How many of you are familiar with Braille? Right, it's the print for visually impaired folks, right? And so you have these kind of like bumps, and you run your fingers across those, and you can read and recognize that. Can you imagine trying to read Braille on the back of your neck? It's not going to work, right? You're like, well, somebody's rubbing a book back there, I guess. I mean, I don't know what it is, but you run your fingers across, and you're like, oh, that's definitely Moby Dick, right? I can read that with the tips of my fingers. So there you go. Cortical magnification. Somatosensory, don't worry about this too much. Object recognition, right? That's pretty cool. Folks it's called haptic perception, right? So you can use touch and kinesthesis to figure out things, right? So you can kind of like, it's always a fun game, right? Like walk around with a box and say, hey, stick your hand in there and tell me what you feel. No, not something. Don't trust people on the street who come up and say that to you. But you can do that, right? You can actually manipulate an object without seeing it, and you can kind of guess what it is, right? How many of you carry a bag that is not see-through? Yeah, right? And so what do you do, Chloe? You stick your hand in there, and you're like, oh, I'm looking for my calculator. Right? And you're like, well, that's a calculator, and you pull out a calculator. You don't reach in there like, I'm feeling for a calculator, and you pull out an orange. Like, oh, no. Right? Because you have pretty good haptic perception. So there you go. All right, let's move on to the vestibular system. Right? Let's try to get through this quickly so you guys have plenty of time for the lab activity. The vestibular system is kind of cool. It involves sort of 3 things called canals, and then a couple things called vestibular sacs. These are each doing similar but slightly different things, right? Your vestibular system in general is giving you a sense of your position relative to the down, right? And whether or not that's stationary or it's whether or not you're in motion, okay? So if you are using your vestibular sacs, the saccule and utricle, that's static, really thinking about which way is gravity, what's my orientation to down, Am I upright? Am I at an incline? Am I lying down on the couch, right?
Some deep sea divers can have, I can't remember the name of it, but they lose track of which way is up.
Yeah.
And they have to use air to identify which way is up. How does that, like, what's going on inside?
Right, so you're in a different sort of media, right? And the density around you is changing, and sort of your sense and feel of gravity is different because your buoyancy is changing, right? And so you're not as sort of sensitive to that. Does that make sense? So normally, like, if you're like walking around on air, you're like definitely getting pulled down, right? But when you get in water, most people sort of float a little bit, right? And there's some buoyancy there, and so you kind of lose that. You lose some of those cues, and you can really get confused and cause some problems.
Plus with like swimming, like most of the time if you want to swim one way, you have to angle your body a certain way to swim that direction. If you're diving and you swim multiple ways, if you don't keep track of which way you started swimming from, you can pretty easily get lost if you can't see the surface. Like if you're cave diving.
Right, right.
Yeah. I mean, it's already going to be dark, so.
Eli, you got something?
Would that have anything to do with like people who have vertigo?
So we'll talk about vertigo in just a few moments, right? That's a great question, so just hang in there. So you've got two of these units in each side of your head, right? Not a big deal. We talked about vestibular sacs, sensitive to gravity. Your canals, your semicircular canals, are sensitive to sort of rotations, right? So as you sort of spin and do your business there, those guys will get stimulated. Don't worry about this too much. Inside of here you have these things called otoliths, which just means ear stones, which are kind of cool, right? I mean, I guess you should start like a heavy metal band called the Ear Stones. I don't know, I thought it was a good idea. So they'll let you know what's going on, so those stones are going to be sort of sensitive to that, right? Basically, you have, if you're rotating, in any direction, that's going to move fluid in those semicircular canals. It's going to deflect some hair cells. It's going to cause those guys to get excited. Because you've got one on each side, whichever way you're rotating, one of them is going to get excited and the other one's going to get inhibited, right? And so you'll know whether you're going clockwise or counterclockwise, or rolling forward or rolling in reverse, right? Because they're each going to be sort of stimulated in different ways, which is kind of cool. This can cause some interesting sort of vestibular system illusions, right? And some problems, we'll talk about those in a moment. But there are sort of four major pathways for your vestibular system, right? There's information that goes to the cerebellum. Cerebellum is really important for monitoring and coordinating movements, right? So if you're going to make some sort of movement, it probably makes a lot of sense to know, if you're upright, if you're reclined or whatever, or if you're like rotating in a circle. We'll also send information to the thalamus. That's just a basic sort of sensory way station there that's going to do a lot of cool processing. It's going to send information to the spinal cord. That makes some sense if you're going to do reflexes, if you're going to make changes in your posture relative to your rotation or angle. And then there's also vestibulo-ocular, right? So that we can control some eye movements as you're spinning around. It's kind of an important thing to do. So it'll stabilize gaze direction. Here is the sort of very complicated process of this. It starts here with your vestibular organs, right? And then you're going to go several places. Here, cerebellum, thalamus, and then out to your eye muscles. Same thing we just talked about, it's just in a flow chart. All right, so let's talk about some perceptual effects with that vestibular system, right? So Eli, you mentioned vertigo. There's vertigo and then there's height vertigo, and often people don't differentiate between the two, but they're sort of important. Vertigo is just, basically you've got some distorted perception relative to gravity, right? So gravity's pulling you down. Your vestibular system is telling you gravity's pulling you at an angle. Your visual system is telling you this is upright. vestibular system says this is upright, you've got a mismatch there that you've got to reconcile that, right? And when you try to reconcile that, that can cause you some serious problems, right? It can cause dizziness, nausea, vomiting, postural instability, and you can kind of like lose color from your skin, right? So you're constantly trying to deal with that. Height vertigo is pretty similar, but it's really involved with that high exposed position, like on a tall building or a ledge, right? And you kind of get that mismatch between the information from your visual system and from your vestibular system. And that's what really causes a lot of these problems. Motion sickness, I don't know if anybody experiences that. It's basically the same idea. It's when one system says, hey, you're not moving, and the other system says you are. This happens to folks when they're in cars sometimes, right? It all happened when they started making cars ride smooth, right? It was really hard to get motion sickness if your car's rattling around or you're not moving that fast, right? But now you can be moving, 80 or 90 miles an hour down the interstate, kind of nice and smooth, right? And so your vestibular system is saying, I'm just kind of sitting still, not really doing a lot. Your visual system says, whoa, man, look at those trees just fly past me. And that mismatch can cause some people some problems. The best thing to do if you have motion sickness is just walk everywhere. You're not going to ride in my car. Hey, I don't know if anybody's seen this happen. Postural alcohol nystagmus, right? This is kind of interesting. Try to avoid acute alcohol intoxication, but I know you guys know people who do this. So the next time you catch someone doing this, one thing you can do is put them down on the couch, which is always a good idea. Not face up, but put them on the couch. And what you'll actually see is one, they'll often develop vertigo, but two, they'll get these involuntary eye movements, right, where their eyes will just kind of like shift over to the side. And again, it's because the information from their vestibular system and their visual system, it doesn't match, and they're really trying to reconcile that, and so they'll kind of like move their eyes around. Alcohol has a really interesting sort of property where it's lipid soluble. It can just slide itself right in to the fluid that's in your vestibular organs, and it changes the density of that fluid. And because it changes the density, it changes the way in which those otoliths move up and down or around and trigger those hair cells, so you get different information than you normally would.
What is acute? Like, what's the difference between acute alcohol intoxication and like stumbling down the path? Because like,
I don't know that there is. I think what this means is if you're like, this is not someone who's like drinking all the time, right, and is just constantly at some sort of
It's like temporary and...
Yeah, but you got to go deep enough, right?
Yeah.
It's got to be, you have to be so intoxicated, right,
Okay.
for this to happen. If you've, just had a beer with dinner, probably it's not going to happen, okay? But you could technically be classified as intoxicated, right, depending on your blood alcohol level and other impairments. But you would have to have a certain amount of alcohol to make this happen. Does that make sense? What about the oculogyral illusion? This is a fun one, right? Everybody could do this right now if they wanted. How many of you have ever spun around in a chair and then you stopped and then you felt like things were, well, like it's moving in opposite directions, right? And so the reason for that is when you're spinning, right, you're getting that fluid moving, and when you stop, right, your visual system says, I'm not going anywhere. That fluid, though, has inertia, right? And so it's still moving around, and so it'll often appear as though you're like moving in the opposite direction, right? Because you're trying to reconcile the visual information with that vestibular information. It's like if you have a bowl of soup and you slide that bowl of soup, that's fine. When you stop that bowl of soup from sliding, what's inside of it continues to move, right? Even though your head's the bowl, the liquid's the soup, keeps moving around, and so you continue to stimulate that vestibular system. But you've actually stopped, so your visual system says nothing's moving, vestibular system says I'm still rotating, and you try to, again, reconcile those two conflicting pieces of information. What's oculogravic? I'm not going to ask you too much about this, but it's when you undergo linear acceleration, right? You'll feel that your body's tilting back a little bit, right? Again, that's because of the changes the vestibular input. Vection is actually kind of cool. This happens a lot if you've ever been sitting at a red light, right? And this particularly happens if there's a large vehicle next to you, like a bus or a truck, and it starts slowly moving forward, and you feel like you're moving backwards, right?
That gives me a heart attack.
Yeah. Yeah, that's called vection, so that's kind of cool. Yes, it's actually pretty impressive. You can do this to yourself all the time if you want. But now you know the next time it happens, like, well, that's just
Less
regular.
interesting. Way to go.
Yeah. I've just, like, taken away mysteries from your life. I mean, there's still that big mystery why you licked a battery, but we're not going to solve it in 16 weeks, so it's a longer process. Now there's my mouse. There's also the Coriolis effect. If your head is moved while you're doing an angular rotation of the body, you feel like your head tilt is occurring. This is what they will often do to folks if they're trying to make sure you can go into space and not vomit. Right? They'll put you in one of those chairs and, like, spin you around and see what happens. All right, questions about that? Comments, concerns? Great. Guess we're the